Okay, well, the recording apparently started. So again, this lecture is about the Hilbert transform, which is described in the last part of chapter 13. And I will kind of start sharing my screen so that we can look at a PowerPoint presentation going through this material. So I assume that by now everybody can see the screen, and this is the start, which is the Hilbert transform. What does the Hilbert transform do? It determines the instantaneous amplitude and phase of a signal. It again is part of chapter 13. It is section 13.6. It's a bit of a weird property or a bit weird property of the Hilbert transform is that it indeed provides the instantaneous amplitude and phase. So in a way it seems almost like it violates the Heisenberg principle of uncertainty because you have instantaneous knowledge of amplitude and phase. As we will see in a little bit, it's not really that because of the filtering requirement, but I'll get back to that. I just thought to kind of burn the name in your brains. It's actually Hilbert, after the famous mathematician Hilbert, and not the Dilbert transform. And by the way, as a disclaimer, I do not necessarily agree with Dilbert's statements in this slide. So, if we kind of immediately maybe give you a preview of what we can do with this transform in neuroscience is that we can find the amplitude of a neural signal, for instance, an LFP or an EEG-like signal, like you see on the right part of this slide. We can also use it to look at the phase of a signal, and of course, if you have multiple signals, like in this example on the right side, you can actually look at the phase relationships between the two. And in that sense, you can then say, oh, who is leading and who is lagging. And that sometimes is pretty useful if you want to analyze signals in multi-channel recordings. Now in chapter 13, there are a couple of these techniques that can do that, as cross-correlation and coherence. And this year I won't have time to go into chapter 14, but there are more techniques that can do that, and in chapter 14 I discussed the Granger causality and the so-called directed transfer function. But for now we are going to look at the last part of chapter 13, which is the Hilbert transform. So unlike what I usually do, I kind of introduce something mathematically. And then we're going to do some examples to kind of show you how things work. In this case, I'm going to go in the other direction. I'm going to explain the Hilbert transform by just taking an example. And then later we go from that example to a more formal mathematical formulation of the Hilbert transform. So now we're going to start a little bit with the math, and again, we are recording this, but that should in no way inhibit you to interrupt me with questions. So it's important that you understand what we are doing, and for that reason, questions may be important. So again, we're going to kind of go for an example, and the example is a pretty trivial example. It's a cosine wave. And the cosine wave has an amplitude of A and a frequency, radial frequency of omega. And we call that thing the signal, ft, the signal. And so what is the Hilbert transform of this particular signal? And I'll explain to you later how we get to that. Well, actually, the Hilbert transform of A cos omega t is A sin omega t, the Hilbert transform. And now if you take the signal, you multiply the Hilbert transform with the complex or with the imaginary number j, Then you get ft plus j times the Hilbert transform, which is indicated as ft with a little tilde above it. Then we get a complex signal, and that complex signal is called the analytic signal. And sometimes if people talk about the Hilbert transform, they kind of forget about this analytic signal, but as a matter of fact, it's basically part of the whole construct. And as you can see, this analytic signal is nothing else than a real part, the cos, and the imaginary part, the sin. And if you take, let's say, for simplicity, a being 1, you can see that we basically get Euler's formula that we have discussed before. And here, in principle, of course, the phase of your signal is omega t, phi, and the amplitude is a, basically a here in both the Hilbert transform and the signal. So how does this work if we try to combine this? And I'm going to project now figure 13.6, and this is again the example that I was just talking about. We're going to take the cosine here, and now we are going to consider the Hilbert transform, which is basically the sine, which is this signal. If I multiply this with the imaginary number j, and I add them both up, then basically I get this analytic signal, which is basically a real part and a complex part. And again, assume for the simplicity that a is 1. And let's kind of take a point somewhere along the time here, let's say the point with the asterisk. That means that for every point in this combination of the signal and the Hilbert transform, I can basically draw a phasor, in this case also the phasor going to this asterisk, that is representative for each of these points. And you can see that the analytic signal in this case, if the amplitude is 1, is just basically a phasor, a line on the unit circle, where omega t is the phase, the instantaneous phase, and where the absolute value a is the amplitude, the instantaneous amplitude of that signal. I'm going to come back to the frequency domain, but as standard is in the book, the yellow one, the yellow part of the picture is time domain, and the brown ochreish part is basically the frequency domain. And you can already see part of it that's known to you, which is we already did that in chapter 6. That the Fourier transform of the cosine is represented here, and the Fourier transform of the sine is represented there. And I'm going to come back to the rest of this figure. So just to write down what the different components are, here you see again the equation for the analytic signal in the time domain, which is nothing else that if you kind of apply Euler's formula like the unit circle multiplied by amplitude a, which is nothing else than a cos omega t, which is the signal, plus j a times sin omega t, which is the Hilbert transform, and that can also be noted like this. So now we can say that if we have the analytic signal, the amplitude is the real component squared plus the imaginary component squared, and then the square root, basically a simple, straightforward computation. And the phi, the phase, the instantaneous phase, is the imaginary component divided by the real component, and then you take the inverse tangent of that. Just kind of as a practical side comment, if we are going to do this in Matlab to simulate what's going on in this situation, we use the Matlab command A10-2, not A10, because A10 basically does not give you all 4 quadrants, the phase for all 4 quadrants, whereas A10-2 does that. So you will see that if we use the inverse tangent function in Matlab, we will use this A10-2 function. Good. I kind of promised you that I would get back to the frequency domain and to the systems analysis in this case as well. And now realize that the Hilbert transform can be actually considered as an operation by a linear time invariant system. So our LTI system that we discussed a couple of lectures ago. And the Hilbert transformer, what it does, it takes the input as input, it takes our function, in this case, in our example, that was the cost function. And as output, it gives you the Hilbert transform, in our example, the sin function. And what is kind of clear when you go from cosine to sine is that you have a phase shift. I mean, if you have the cosine, it starts at 1 and then it keeps oscillating. If you have the sine function, it starts at 0 and it keeps oscillating. But the functions in themselves are the same. If you shift them by 90 degrees or minus by 2 radians, then you get you can kind of shift the cosine function into a sine function. And now I get to the more generic point of what the Hilbert transformer is, because that is actually precisely what a Hilbert transformer is doing. It takes a signal, it kind of takes that signal and shifts the phase by minus 90 degrees or minus pi over 2 radians. And if you do that in the unit circle, it means, and I'm just pointing at it now, it means that you are multiplying it by the exponential minus j over pi over 2. And if you do that, you can see that that actually corresponds to a multiplication with minus j. So as a matter of fact, it's a pretty simple operation if you think of it like that. You just take your signal, you're multiplying it by minus j, and then you get your Hilbert transform. And if you don't believe me that that is generically true, you can actually read that part in the book. There I just take a kind of a random complex number, and I apply that multiplication with minus 2 to that complex number, and we find indeed that that results in the minus 90 degree shift. So if that is the case, let's go back to that figure that we looked at a few minutes ago, where we have our signal, where we have our Hilbert transform, and where we have the combination in the analytic signal and the representation in the Argand plane with the real axis and the imaginary axis as a phasor on, in this case, if A is 1, the unit circle. Then basically I can do the same thing for the Fourier transform. I already determined that this was the Fourier transform of the cosine. This is the Fourier transform of the sine wave. And what we now have to do is we multiply this thing with minus j. And if we do that, we get a magnitude multiplied with minus j. To keep it simple, or to keep it a little bit generic, we also do this, of course, with the negative frequencies. But because the negative frequencies have a negative omega, I can basically consider multiplying that amplitude number with j instead of minus j, basically to correct for the negative frequency. If you do that, and you can follow it step by step in the book if you want to. I'm not going to bore you with that type of algebra at this point. Then what you will get is that the negative frequencies will cancel, and the positive frequencies will basically enhance each other. And so that means that the Fourier transform of the analytic signal does not contain any energy in the negative frequencies, but it doubles the amount of energy in the positive frequencies, which is kind of an interesting fact, and we will see later in the simulation that that is indeed the case. So now let's kind of go to this signum function. The reason why we kind of use this signum function is that we multiply the magnitude with minus j if the frequency is positive, and we basically, so when signum omega is plus, and we multiply with plus j if the omega is negative. In other words, if signum omega is negative, and then negative j, of course, is positive j. That's the signum function. So signum function is just either 1 if omega is positive, or -1 if omega is negative. Another thing to remind you, because recall that 2 slides back I showed you that the Hilbert transform is nothing else than a linear time invariant system that is shifting your signal with minus pi over 2 radians, or minus 90 degrees. And so kind of let's have a look again at the generic rules for an LTI system. We had determined that if you have an LTI system and you give it a unit impulse, delta t, that you get out the so-called unit impulse response, or HT. And we said, well, if we have an LTI system, that's really all you need to know, because these linear systems are pretty simple, although simple in between quotes here, because they can become pretty complicated analytically. But simple in the sense that they obey the simple rule of scaling and superposition. That means that if I have some input, xt, and some related to that, some output, yt, of the same system, that basically I know yt as the convolution of xt, which is my input, with ht, my unit impulse response. Hence, you only need to know this thing to kind of compute the output for a given input. And recall again that we then decided to kind of look at that in the frequency domain. And there, lo and behold, we found that that complex convolution operation, which is an integral operation, is actually pretty simple in the frequency domain, because if you do a frequency transform, a Fourier transform of xt to xj omega and ht to the frequency response hj omega, then you just multiply the two and you get the Fourier transform of the output. So that makes life a lot simpler in this particular case. Now keep this in mind, and now we will basically use this construct to get a more mathematically formal description of our Hilbert transform. So this is basically giving you the overview. So let's kind of take a little bit of time to look at this. There are many aspects in this, but in the end we are getting here the mathematical formulation. And I do not expect you to maybe immediately understand all the details. So basically, if you need to look up some details later, you can always read page 277 to page 280. So let's go back to what we had. We had an Hilbert transformer that transforms some sort of a signal into its Hilbert transform, ft to ft tilde. And recall that in the frequency domain, we just kind of discussed that, we have a minus 90 degree multiplication, and that is basically nothing else than multiplying the complex number with minus j. And also recall that for the negative frequencies, we have to do that with plus j. And that leads to the fact that we now have an hj omega, which is kind of what we need to do to multiply the input with in the frequency domain. Hence, that must be hj omega. Let me go back. See, if you, in the frequency domain, if you multiply your input with something to get the output, then that multiplication factor must be hj omega. So our hj omega, in other words, is nothing else than that multiplication with minus j corrected for the sine of omega. And so this is the minus j signum omega. So now the question becomes, what if we look at the inverse Fourier transform of this, what do we get for the unit impulse response? Because if you take, let's say, the frequency response and you do the inverse Fourier transform, you must get your unit impulse response. Look, if you have your frequency response and you actually go to the time domain, you will get your unit impulse response. So knowing this, we just kind of use this, and I fully admit you can be lazy here. You can just go to a table and find out what the inverse Fourier transform is. If you do that, you will find that it's 1 over pi t. And so that's basically, if you wanted to characterize this system, you can say, well, ht is 1 over pi t. That's it. Which now means that if I have some sort of a function ft that I want to convert to its Hilbert transform in the time domain, I can do a convolution of ft with 1 over pi t. And recall that if you do that, you will get this equation. For now, kind of ignore the CPV comment there. Just kind of read around it, and then you see 1 over pi, the integral ft divided by t minus tau, d tau, is just the standard equation for the convolution. Now there is a bit of an awkward situation here in this integral, and that is that for t is equal tau, this thing goes to infinity. So in other words, there is a pole within the integration limits. And that is exactly the reason we have here the so-called Cauchy principal value operation, the CPV, because that's a little mathematical trick to get around the integration with a pole in its domain. And again, I'm not going to go into detail of that. If you want to, you can read a little bit about that in Appendix 13.3. And if you want to kind of know a lot more about it, then I refer to a couple of textbooks. For now, it's kind of not so interesting. Forget about the CPV and think about the fact that it is a convolution, and you have to just use that CPV to get around this trick, or to get around this problem of having a pole within the integration limits. So now I think we're ready to take our example and go to a numerical implementation in Matlab. And if you want to run it, you can run it. I'm going to simply indicate to you in this listing what the different components are in the Matlab routine. And the program that we are using is program 13_5. And the part that I want to point at is that we're using the same example as in our figure. We're using a cosine signal as the signal. It's 4 Hertz, as you can see, 2 pi 4, and so 2 pi 4 is omega, and the 4 is the frequency in Hertz. And now you can see that we are using the so-called Hilbert command in Matlab. And keep in mind, the only thing that Hilbert command is doing is multiplying with minus pi over 2 radians. And what it does is, as a matter of fact, in that sense, I should correct myself. It's a bit confusing maybe, because the Hilbert command in Matlab does not produce the Hilbert transform, but it produces the analytic signal. So it's basically the signal plus j times the Hilbert transform. So they should have called it analytic instead of Hilbert, but well, it's above my pay grade. So now we can use the equations that we defined. We can say the instantaneous amplitude, which is here, is the absolute value of the analytic signal. And the phase, and here you see what I was saying, we're using A10-2 for the inverse tangent. And we kind of take the imaginary and the real components of the analytic signal. And then if you, of course, if you basically differentiate your phase, then we get the instantaneous frequency. And then this part is plotting that data. And here you can see your cosine. You can see the phase of the cosine. You can see the instantaneous amplitude of the cosine. And you can see the instantaneous frequency of the cosine. And as you can see, there is no big surprise here. This is all pretty much what you would expect. Then here I then go and compute the FFTs of both the signal and the analytic signal. And basically I plot here in this case the absolute values of the Fourier transform. And here you can actually see, the other part is not very visible. Here you can see that there is only energy in the positive part of the spectrum, just as in that figure that we discussed. And then the last part of this program is looking again at the imaginary part of the analytic signal, which is basically plotted in red. And that should be the Hilbert transform, i.e. the sine wave. And see in the red, indeed, we do have a sine wave. And then we plot basically two things. We plot here the real part of the analytic signal and the signal itself in different colors. And again, you cannot appreciate that here, so run the program yourself in Matlab, where you have the programs. Then you can see that they basically superimpose, which is what you would expect, isn't it? The real part of the analytic signal and the signal itself should be identical. So that's basically a numerical example of the example that we discussed in this figure here. We have the cosine as the signal. We have the Hilbert transform here, the sine wave, and we have the combination of both being the analytic signal. So before I go into details of some applications, are there any questions about this explanation, the example, and the underlying mathematical equations? If not, then we're going to go to a couple of examples. First of all, I'm going to give you an example that one of my students a long time ago produced. And then we are going to go to a couple of examples where we are currently working with analyzing data from the neonatal ICU. So we can use, I kind of mentioned that before, we can use the Hilbert transform and its capability to detect amplitude and phase in an instantaneous fashion. We can use that to look at connectivity and synchrony in networks. We can use it to look at amplitude fluctuations across signals. And it's not unimportant because, quite frankly, I would say that one of the important frontiers in neuroscience at the moment is actually understanding networks. We know quite a bit about individual neurons. The mathematics of that was figured out quite a while ago, the fundamental component in 1952 by Hodgkin and Huxley. And nowadays we know more and more about all these channels. So in that sense, we start to understand individual neurons better and better. The part that we really struggle with as neuroscientists and neurologists is understanding networks, the connectivity and their function, and how they synchronize and desynchronize, and what the meaning of that is. I want to kind of point out And the first figure of the chapter we are not going to discuss, chapter 14, but this figure basically drives home the issue that a lot of people do these phase analyses, and they do analyses of correlation or coherence, and then they derive more or less the connectivity, the network connectivity of that. And of course that cannot be really done, because if we find that neuron A and B have similar activity but that there is a delay delta t between them, you cannot just say that there must be a connection between the two. That might be, but it might not be the case. And very trivial reason is that, well, if you have let's say two neurons with a delay in between there, it could very well be that there is a third neuron that is causing the activity in both. And if delta T1 is not equal to delta T2, then B and C are not going to happen at the same time. Delay. But as you can see in this case, there is not really a connection between it. So you have to be careful with all these analyses, and there is actually no mathematical analysis of functional data that can show you connectivity. They can only kind of give you an idea of putative connectivity, or if you want to be more careful, you can say it tells you something about the synchrony, the level of synchrony or asynchrony. One of the things we did, again, a long time ago, Amber Martel, at the moment Amber Nolan in my class, she's now in San Francisco, moving to Seattle, still working as a neurologist, scientist in neuroscience. And she did some interesting work on looking into seizure models in isolated networks. And what she did to do that is she recorded from single neurons intracellularly, and this is an example. And she recorded at the same time extracellularly in the surrounding. This is an example as well. And these recordings were made simultaneously. And at some point we were interested during different scenarios, what is the lead and the lag, and what is the level of synchrony. What happened at that time is that we first kind of made some sort of an activity converter, and so we kind of changed this into the spike raster, and then we converted that activity, the spike activity here and the spike activity there, into a continuous signal that represented the frequency of the underlying neurons. And you can already now see a little bit better how they agree and disagree. But what we then did is we then filtered it and then did the Hilbert transform. So basically, we then got a phase, an instantaneous phase of the activity of the intracellular activity. And we got the instantaneous phase of the activity that we recorded on the extracellular space. What we then did is we subtracted the two. So now you have the phase differences between the network and the individual neuron that's embedded in that network. And then we looked at the distribution of the phases. And of course, if the phase distribution is 0, it means that they are perfectly synchronized. If it's negative, it means that one leads the other. And if it's positive, it's the other way around. So one is lagging, one is leading. And so what we did in a number of cases, and if you want to read the details about that, I refer to the paper in the book. But you can see that for every combination of network and cell, we kind of then in the end we got a histogram of phase differences. And in this case, we could conclude that this neuron was basically lagging a little bit behind the network on average. And so in that sense, we can use the Hilbert transform to get an idea of the relationship of networks and cells that are embedded in that. And then the last example we're going to talk a little bit longer about is the example that Sylvia, the student in my lab, has worked on together with the neurologist, Dr. Henry, and basically the other Dr. Henry, although one Dr. Henry is the last name, and the other Dr. Henry is the first name. So it's Dr. Henry Square, I think, in this case. But Sylvia, why don't you get started, and I will kind of give you the first slide so that you can give a little bit of background of the rationale and the motivation of this PhD project.
Awesome. Thank you. Hey guys, my name is Sylvia. I'm a first year in Vim's lab and a second year overall in the MD PhD program. I'm going to talk to you, like Vim said, briefly about neonatal seizures, so you get some sense of the practical application behind signal processing applications like the Hilbert transform for seizure activity. So as you guys were introduced to last week, seizures are classified as abnormal hypersynchronous activity in the neurons of the central nervous system. And neonatal seizures specifically are those that occur in the neonatal period, which is within the first month of life. So as you can see in this graph on the slide that Vim has up, seizure burden is highest actually in the neonatal period. And part of this penchant for heightened seizure susceptibility might be factors that are intrinsic to infant brains, but they're extrinsic insults to the central nervous system that occur during the neonatal period that usually end up manifesting as seizure activity. So if we go to the next slide, we can see here, this chart shows that the majority of neonatal seizures are symptomatic. And so what symptomatic means is that these seizures are a result of some type of extrinsic factor that leads to acute central nervous system insult. And because of the resulting brain damage and limited central nervous system development in most neonates, the majority of infants end up having subclinical seizures. And a study by Scher et al. Puts that number around 58%, and there's been subsequent studies that have had similar findings. And subclinical, what that means is that their seizures cannot be detected visually through established clinical markers of seizure activity. So there is a huge reliance on EEG for accurate seizure detection. So if you go to the next slide, to the next one. Yeah, we can see here that accurate detection is really, really important because it's important for guiding clinical management, which includes diagnosis, treatment, and prognosis. So this is especially important when we're looking at neonates who have, because they have a high amount of seizures in a short time span. And so since studies have shown higher overall seizure burden is associated with more adverse neurodevelopmental outcomes, it's really important for us to have robust detection that can tailor treatment to prevent over or under treating these infants. Currently the gold standard for seizure monitoring and detection is continuous EEG, but there's also efforts to create simplified automatic detection algorithms that can more easily identify seizure activity and identify trends in brain activity. And this is of interest to my project and Vim's lab, which is why today, in the next few slides, we'll be showing you how the Hilbert transform can actually be applied to actual data from the neonatal ICU to extract the instantaneous phase and the amplitude of the signal. Next slide.
So now I will take over again. Thank you, Sylvia. So what you typically try to do if you try to find analysis technique is that you first simulate a case that is relatively ideal, and you see if your technique works. And that's kind of a generic approach that I can recommend to all of you, that if you come up with an idea to analyze your data, don't immediately jump into it. Just kind of make a mock-up data set that is pretty much ideal and see if whatever you have in mind is actually working. And if it's not working on your idealized data set, you can pretty much forget it will work on real data that's really noisy. In general, you can almost say that it's maybe not 100% true, but it comes pretty close. So what this slide is showing us is a recording with 10 channels. And each channel contains a bunch of noise. So you can see that black noisy signal here in each of the channels. And what I did is I just added let's say a simulated seizure, which is kind of a 2 Hertz sine wave with different amplitude in channel 1, a little smaller one in channel 3, and then a little bigger one in channel 6. And kind of assuming that, okay, this is 10 seconds or 30 seconds or whatever. No, it's actually more. I think it's 80 seconds of recording here. This is the calibration, if you want to know. And the amplitude, since it's a simulation, of course, is in arbitrary units. You can see that basically, yeah, you have here a recording with that's pretty noisy with some high level activity. The question is, in this really idealized and simple case, can we apply the Hilbert transform to make sense out of this? So the first thing we need to do, and that is what I promised all in the beginning that I would come back at, is how can this basically pseudo-violate the Heisenberg uncertainty? Well, the first thing you do is you have to apply a very narrow band filter. Otherwise, the Hilbert transform is going to give nonsense. And so what I did is this black signal that you see here was filtered with a very steep filter from 0.5 to 1.5 Hertz. And the reason I did that is because I know that my frequency is around 2 Hertz, or is exactly 2 Hertz in this case. When you do that, you get the red signal. And you see that I superimposed the red signal, the filtered signal, on top of the raw black signal. And now I'm using the filtered signal in this example to determine the instantaneous amplitude and the instantaneous phase. And that can be seen in this right panel. If you look at the top 10 traces, I'll get back to this trace here, the lower trace. You can see that I repeat the red signals here. And superimposed on the red signal, I plot in green the instantaneous phase and in blue the instantaneous amplitude. And you can see that indeed the instantaneous amplitude is very nicely following the amplitude here of the seizure. And here the seizure is smaller and the amplitude reflects that. And here the seizure is larger and the amplitude reflects that as well. And then for each of the channels, we had also, of course, the instantaneous phase. Now, what we were thinking, and we can already see that this is not going to work very well, is that we might also use the instantaneous phase to kind of see if there was synchrony across the channels. And the simple thought was that, well, if in the noise the phase of the channels is pretty random, when you start averaging these phases, they could kind of should end up being close to 0. And if during the seizure the phase is not random, then during the seizure the average phase should be clearly much more reflecting that synchrony. And you can actually, we're going to do that for homework, not this week, but for week 6. You can see that indeed to some extent that's true, but it is really it's really not a very clear outcome of this average phase. It's not like, oh yeah, it's completely random here and it is absolutely synchronous here. Due to the noise that's still residual noise that's still there, it's not a very clear-cut difference. Any suggestions why that? How we can explain this failure? Just think back about your signal averaging. You know? So what is in the end, what does determine the quality of your average? Well, maybe I'm going to leave that for a homework question then. It might be good to think about that. There is really, in that sense, no difference between the average of stuff in the time domain or stuff like this, like that's already in a derived factor such as the phase. So we then thought, well, let's kind of apply this to a real signal, and that's basically this slide. And what you can see in this slide is a recording of 3 signals. They are, if you recall, this one is the 10-20 position on the frontal. This is a central 10-20 position, and this is a posterior occipital 10-20 position. And we are dealing with 3 signals, and they are already filtered here. These are the red traces. And this is a pretty long recording so that you can get some perspective of the context and the surrounding of that seizure activity. What you can see here in this case is that in each of the channels we have the blue signal, just like in the other example, the simulated example, which reflects the instantaneous amplitude. And we have the green signal, which reflects the instantaneous phase. And just as in the simulation, we do have an average phase here. And just as in the simulation, of course, there is not much that we can do with this signal. So sometimes actually useful to falsify your ideas. So, well, great idea, but it doesn't work. And, that's probably true for many ideas that you might have in the future as well when you do your own research. What does work is the amplitude, of course. We can see in FP1 that the amplitude is gradually increasing, and the blue line. This one is gradually increasing, decreasing, and increasing again. And this one is a little bit less clear until you get towards the very end of the recording in terms of the amplitude. Now, the neurologist is a very refined pattern detector. That's the reason we call the judge of the neurologist the gold standard. And the neurologist basically independently, completely blind to what we were doing, found out that the seizure was starting here. And if you want to see what this looks like in detail, this is how this was picked up. This is the raw signal. And here you can see a couple of bursts, and here you can see a couple of discharges. This is the same signal, but now filtered, basically as the same as the red signal here. And this is the place where the neurologist marked the seizure onset. I think the neurologist is still online. Do you want to comment on that, Julie?
Sure. I don't remember exactly which seizure this is that we picked out, but we're using, or at least personally, when I'm reviewing an EEG, I'm looking at it qualitatively and then can bring in some quantitative measures to, see if my suspicions are correct about an onset of a seizure. But really, it's just pattern recognition. So I'm not looking at the individual amplitude or the individual frequency or morphology and kind of consciously going through it piece by piece. It's more of a, you know, pattern that you can intuit after reading a lot of EEGs.
Yeah, it's not something that you pick up probably after having looked at 10 EEGs. You have to, you're talking probably years of EEG. And then in addition to that, the EEG of these neonates is very different from the EEG of an adult or a kid that's a little bit older. Well, thank you. I think this this concludes this example, and also my my introduction to the Hilbert transform. So we can kind of wrap up with some questions and some comments. So that means that if you want, you can stop the recording. I hope the recording went well, but we'll see. If the recording went well, we make them available to you.
Well, thanks so much for inviting me to this lecture. I've got to hop off and go see a patient. But if there are any...
Thank you for joining.
Oh, you're welcome. Happy to. Bye.
Bye.
William, did you say to stop the recording?
Yes.
Cool.